Welcome to the Chem 116 lab dilution video featuring the group comprised of Kapil, Cecil, and Eliza. In terms of safety, all group members were required to wear the appropriate goggles as well as closed-toed shoes and long pants. For the procedure, to begin, the group acquired and cleaned the necessary materials in order to utilize them later on. The group washed the Moore pipette by flushing it 3 times with water and 3 times with a blue solution to get rid of any remaining water that may impact the readings. Challenge 1 presented to the group was to determine the density of water using a 10 milliliter graduated Moore pipette and a 100 milliliter graduated cylinder. The group decided to fill up the 100 milliliter beaker to 100 milliliters and use the Moore pipette to systematically pipette water out of the beaker and into a cylinder, and check the mass of the beaker in order to find the density. Challenge 2 was to prepare a series of sugar solutions and measure the density of each solution. The group decided to use the volumetric flask and the stock solution. The group poured the stock solution into a graduated cylinder and then used the Moore pipette to pipette the solution into the 25 milliliter volumetric flask and then add water to it. Challenge 3 was to determine the density of an unknown sugar solution. The group decided to weigh the 100 milliliter beaker alone and then add the sugar solution to find the density. The group first decided to weigh the mass of the empty 100 milliliter beaker, which measured approximately 83.059 grams. Challenge 1, the group decided to utilize the graduated cylinder, the graduated pipette, and the beaker. The beaker was first filled up with 100 milliliters of water, and the mass of the beaker with the water inside it was weighed on an electronical balance. Water was then systematically pipetted from the beaker to the graduated cylinder, and the mass of the beaker was weighed after each pipetting using the electronical balance. The volume was told by the meniscus and both determined density. The definition and the usage of a meniscus come into play here. A meniscus is a curve in the surface of a liquid when it touches another material. They can come in two types, convex and concave. A convex is shaped like an upside down U, like when mercury is found in a test tube. Concave, the meniscus is shaped like a right side up U. This is most common and is found when water is present in a test tube. Adhesion is the scientific phenomenon responsible and is defined as the joining of two different substances due to attractive forces that hold them. Adhesion is responsible for a meniscus because water molecules are attracted to the molecules in the wall of the glass beaker. And since water molecules like to stick together because of hydrogen bonds between each water molecule, when the molecules touching the glass cling to it, other water molecules cling to the molecules that are touching the glass, which forms the meniscus. A concave meniscus can be read by locating the lowest point of the curve and reading the instrument to 1 degree of uncertainty. For example, if the ones place is given on a pipette, we should read the meniscus to 1 decimal place, or the tenths place. All group members performed at least one dilution. To complete challenge 2, the group decided to utilize the 25 milliliter volumetric flask and the stock solution. The group first poured the stock solution into the 100 milliliter beaker and then, using the Moore pipette, systematically transferred amounts of that stock solution into the 25 milliliter volumetric flask, then filled the volumetric flask up to the mark with water. This allowed the group to utilize the C1V1 equals C2V2 equation of dilution. For challenge 2, the group utilized stock solutions and dilutions. Stock solutions are concentrated solutions that can be diluted for future laboratory purposes and use. Dilution is defined as making a solution less concentrated, usually by way of adding more solvent. Solute is the substance that's being dissolved, in this case sugar, while solvent is the substance that is doing the dissolving, in this case water. The formula for dilution is C1V1 equals C2V2. Using algebra, we can find the concentration of any one variable if we know the other three. All possible combinations and permutations of each of the variables can be found on the picture on the right. C1V1 is the initial concentration. V1 is the initial volume of solution, or the volume delivered. C2 is the final concentration of the newly diluted solution, while V2 is the final volume of the newly diluted solution. Using the C1V1 equals C2V2 equation, the group was able to find the concentration of different sugar solutions in the volumetric flask. The group poured approximately 100 milliliters of the unknown solution into the beaker to measure the mass of the beaker plus the solution to find the density. Upon completion of all challenges, the group cleaned up their area, disposed of any excess reagent down the sink, washed their hands, and turned in the worksheet as instructed. During this experiment, the group learned a lot about dilution and stock solutions and how to use them in the laboratory. Thank you!